All right, so this is a session called Downstream, but as I say, some of it is looking at antifibrotics and anti-inflammatory medicines, but Joanne is going to talk about Edgewise's approach, which is not actually downstream. It's looking at muscle fibers and strengthening them. So, but this is not dystrophin expressing in terms of antisense, oligonucleotide, or gene therapies. So our first speaker is Linda Marban from Capricor.
Okay, I always wanted to be a jazz singer. Here I go. This is my big chance. I won't sing, I promise. I'm Linda Marban, CEO of Capricor. We are a NASDAQ-listed, publicly traded company, and all of our filings are up to date with the SEC, so have at it if you are interested. Today I'm going to provide a brief overview of CAP-1002 and our progress in Duchenne muscular dystrophy. There's a lot of background noise, so is it bothersome to anybody but me? It's okay? All right, CAP-1002 is not a stem cell therapy. It is a stromal cell population that works by releasing exosomes, and exosomes track decisive injury, releasing a variety of nucleic acids that drive activities that are both immunomodulatory and pro-regenerative. We have over 100 publications on CAP-1002, so please feel free to make use of those to understand the mechanism of action of our very powerful therapeutic for Duchenne. The nice thing about CAP-1002 for patients and families is that it is a very simple procedure. It is a 45-minute infusion. Ultimately, it can be done on an outpatient basis and with very few side effects. And I'll talk about safety in a few minutes, but it's very safe. It's been in over 200 patients over time, and with a simple pre-medication regimen, we have almost no issues treating patients. I'll just briefly remind you of the data from the HOPE-2 clinical study. It was published in The Lancet last year, where we saw a 70% delay in the decline associated with skeletal muscle performance as identified by the performance of the upper limb. That means the patients that were treated with CAP-1002 had a very significant prevention of progression of the disease. We also saw improvements in ejection fraction, which is how the heart meets the needs of the body. And this is probably the first time that there's been a statistically significant improvement in cardiac function in Duchenne cardiomyopathy. So we are laser focused on following up on this. So I'm going to talk to you about the open label extension results today, 24 months. Without further ado, as I mentioned, we did the HOPE-2 clinical study. For capital reasons, that study was stopped early. By the time we were able to get the open label extension started, it was an average of a year off of therapy for our patients. We now have treated all of our patients that came back into the open label extension, whether originally placebo or CAP-1002, with 2 years of continuous infusions, and they are now almost all of them continuing into the third year, and some have even asked for a fourth year, because what you're about to see is very significant clinical benefits. Most importantly, it's safe. With the exception of some annoying headaches and a little bit of infusion-related issues. Patients do really well on this. There's been no treatment-related serious adverse events that have slowed anybody down longer than just a little bit post-infusion. So let me go through this data with you. I don't know if this has a pointer on it, but what I'm going to show you here, if you can look at this figure and divide it in your mind into 3 sections. The first section is the HOPE-2 clinical study. The dotted line phase is the off study time, the gap phase we call it, and then the second 2 years, or the second half of the graph, is the open label extension patients. In blue, you will almost always see in my figures, those are the patients that are on CAP-1002. In red, those are placebo or non-treated patients. And what you can see in this very important figure is that if you were lucky enough to be randomized into receiving CAP-1002 at the beginning of the HOPE-2 clinical trial, you are much better off 4 years later. So this is evidence of what we would call disease modification. That means that even when you get on CAP-1002, your disease decline is significantly slowed, and therefore you have a greater opportunity of sustaining muscle function. And for our patients, this is measured by the performance of the upper limb, shoulder, arm, and hand strength. And what's interesting is we published that there was a 70% delay in disease decline, and in the open label extension, we are seeing a 66% delay in disease decline, so consistent performance across multiple years of study. So this graph is a little complicated, but I like to show it so it identifies for you that once you're on CAP-1002, wherever you started off in the world, you are better off several years later. So in blue, in the dark blue, are the original CAP-1002 treated patients, and in light blue are shown 2 years of the original placebo group. And you can see over the course of the open label extension study, there is no difference between these two patient populations. So time is muscle, guys. Once they get on CAP-1002, disease progression is significantly attenuated. Shown in red is for your reference, that is the first year of the placebo, and then in dotted line is the second year off of treatment for the placebo patients. What's interesting is our placebo patients decline just as natural history has predicted that they will, which is about a 4-point decline per year, and that's been published by Anna Mayhew and colleagues, as well as Marika Payne and the group in Italy. So very significant improvement in disease progression, and that's still significant years later. So let's talk about the heart. I think everybody is aware of the fact that one of the major life-altering issues in Duchenne is the cardiomyopathy. Nearly every person with Duchenne has implications of cardiac function or dysfunction by the time that they are 18 years of age. This has been published and well studied. We still are working towards getting a therapeutic specifically directed towards the heart disease associated with Duchenne. Right now, the only thing that's available is sort of standard of care heart failure medications for patients. And for the first time, we were able to see perhaps that there's an attenuation of the cardiomyopathy by CAP-1002. So let me just briefly go through this data with you. Shown in the gold or yellow line at the bottom is a natural history data set, nicely made accessible to us by Cincinnati Children's Hospital. Over 2,000 MRIs are aggregated there, and it shows that there is an annual average decline of 0.78 points, or almost 1 ejection fraction point per year over time. In our open label extension study, now we didn't collect MRI at baseline. We didn't want people who had gone through a clinical study to have to come back in for getting in the magnet. So we decided originally just to study the performance of the upper limb, but the performance of the upper limb data was so good that we wanted to see what was happening with their hearts. And what we were able to see is that after 24 months on continuous treatment of CAP-1002, so 8 infusions, 6 out of 9 patients had improvements in ejection fraction. Now, guys, people don't have ejection fraction get better in Duchenne muscular dystrophy. So we're not saying that this is obviously approvable data, and what we are saying is that this is very important to take a look at, to acknowledge that ejection fraction improvement is not usually seen in Duchenne, and also very importantly that we see a direct correlation with the performance of the upper limb. So the ejection fraction, which is not performance driven, can be used to predict what's going to happen potentially with the performance of the upper limb, skeletal muscle function, very important for the regulatory pathway. And then finally, I just want to say, everybody says it's become the colloquial expression in Duchenne, time is muscle, but you saw from our data that if you happen to get on CAP-1002, the earlier you get on, the slower your disease goes. That's what we want for all of these people, whether they are receiving a gene therapy or any other type of therapy. The importance is to maintain that functional improvement, and CAP-1002 can do that. We are recruiting our HOPE-3 trial now. We're over 50% enrolled. We are laser focused on finishing this trial in our later stage non-ambulant patients so that we can get approval and then hopefully indication expand and get these kids as early as we possibly can. And as I mentioned, and I want to emphasize, whatever your sons are on, or you yourself are on, CAP-1002 is a good partner in the sandbox because it'll help delay the decline of the disease, help to preserve the function that you have. And so with that, I just want to highlight that we have a great partnership with NS Pharma. We selected them specifically. They selected us specifically because they have Viltepso. It's an exon skipping drug. They know all about Duchenne. They have a great team here in the United States. They have rights to market and distribute our product in the United States and Japan. We do everything else up to that. So if you're interested in HOPE-3, which I hope you are, please visit our booth outside, contact our clinical team. We have 20 sites across the United States that are actively enrolling at this time, and let's get this trial done and let's get CAP-1002 approved. And finally, I just want to thank everybody that makes this study possible, but most importantly, the patients and the families. And thank you, PPMD, for always being there to drive this program forward and all the programs involved. And I will turn it over to the next person.
All right, thank you, Linda. Our next speaker is Joanne Donovan from Edgewise.
Thank you. I have to look. I'm sorry. Where it is. Okay. Great. Thank you. I'm very happy to speak with you today, and thanks to Parent Project for the opportunity. So my name is Joanne Donovan. I'm Chief Medical Officer at Edgewise, and I'm going to speak to you today about EDG 5506, which is an agent to preserve and protect muscle in Duchenne muscular dystrophy. We have a forward-looking statement because we're a public company, and most importantly is to note that EDG 5506 is an investigational agent and is not approved in any territory. So the fundamental approach that we are using is to protect susceptible muscle fibers. We know that some muscle fibers are more susceptible to damage because of the lack of functional dystrophin. And this is, as you heard from Anamika, the fundamental problem. When you don't have dystrophin, the muscle can't protect itself from damage. What we've done is make an investigational therapy, EDG 5506, that's designed to protect these susceptible muscle fibers from damage, regardless of the mutation. What we've seen is that in diseased animal models, like the MDX, EDG 5506 protects susceptible muscle fibers and prevents long-term development of damage. Now, going to the basic problem here, what happens to dystrophic muscle that it breaks down, that it isn't protected? And what is, I don't think I have a, I can't show you with a laser pointer, but what we have on the slide is at the top a schematic of muscle fibers, and they're tied together by the green proteins, essentially, which is dystrophin. So that when a muscle contracts, there is only a subset of fibers that contract in the middle panel. And what the dystrophin does, it allows the force of the muscle fiber to be spread over the other muscle fibers. So it spreads the contraction-induced force, and it prevents damage. Without dystrophin, contraction causes injury. That muscle fiber takes everything on itself. So what we're trying to do is to basically take the edge off that damage and prevent the contraction-induced damage. What the next slide shows you is actually in a mouse model, the MDX that you're familiar with. And this is a finger muscle of a mouse, and it's so small that you can see through it. So this is in response to contraction. And on the left, what you see is that you start to see these little blebs pop up. And that's actually muscle fibers breaking down with maximal contraction of the dystrophic muscle. So what you see is that contraction causes damage to the muscle. You're starting to see these fibers break up. And if you pre-incubate the muscle, if you're treating it with EDG 5506 on the right, the muscle contracts. It retains function, but you don't have this contraction-induced damage. So really going to the fundamental problem in muscular dystrophy, to be able to protect the muscle. Now, what happens when these muscle fibers break up like this on the left is they release biomarkers, things like CK that you've heard of, and other biomarkers of muscle damage. So we're able to see that in the preclinical models. We're able to see that in the clinic. And so far, we have looked at patients with Becker muscular dystrophy for over a year now, and we see those biomarkers of muscle damage go down early, rapidly, and in a sustained fashion. So an 80% decrease in the fast muscle troponin that is really the target of EDG 5506. And along with that, we actually see a trend towards improvement in these men with Becker. So what we've done is now moved into looking at initial phase 2 trial in boys living with Duchenne. We're looking at about 27 boys aged 4 to 9 up to their 10th birthday who are ambulatory. They're on chronic corticosteroids for at least 6 months. They can be on exon skipping therapy as well. We have a 3-month placebo control period because, again, we saw those biomarkers go down early and stay down. So we can use this to be able to choose a dose to understand the pharmacokinetics in boys and be able to choose a dose for the phase 3 study. This is followed by a 9-month open label period where everybody gets EDG 5506, and we're actually extending that open label beyond the full year of this study. The investigations that we're doing in this study, there are about 10 visits over the year. It spreads out so that it's every 3 months, and then when we get to past a year, it'll even be less frequent than that. It's an oral drug taken once a day at home. We're also looking at at-home elements to minimize having to come to clinic with things like home functional assessments as well, accelerometry to look at the activity around the house. We're looking at things like echo to monitor the heart, DEXA to look at muscle mass, and the usual functional endpoints that you're familiar with in clinic. We are looking at a number of sites across the country for this study. And what this study will give us is the information to start a phase 3 study. We are, to summarize, this is a drug that is taken orally. The intention is to preserve, to protect the muscle and allow it to regenerate, so potentially improve, as we've started to see in the Becker patients. We are looking at patients with any mutation, so across the spectrum of dystrophinopathy, which is why we're looking at both Becker and Duchenne. To protect the most susceptible muscle fibers, as Anamika said, dystrophin isn't it isn't creating muscle, it isn't contracting tissue, it's there to protect. And so that's what this is going after. It has the potential to be used alone or in combination with other therapeutic approaches, exon skipping, which we're already doing, gene therapy that we intend to do in the future. And really to go to the fundamental problem to stop the damage where it begins. And I think I forgot the last one, the last time. We can be contacted. We have a booth out there to ask us questions and get in touch on our website. There's a link to the clinical trials and can get more information there. So thank you, as always, thank you to the parents, the families in the trial. We know it takes a lot of effort to be in these trials. So we very much appreciate it and want to move things forward for the community. Thank you.
Thank you, Joanne. And next we have FibroGen.
Great. Thank you, Pat. First of all, before we get into FibroGen and who we are, I wanted to say a special thank you to the patients, the parents, caregivers, and the community for participating in all our clinical trials. So thank you for that. Without you guys, we have no studies. We can't do what we're doing. So my name is Akshay Patel. I head up the field medical. And at FibroGen, we were founded about 30 years ago. We're based in San Francisco, and we're about 600 people worldwide. And I think most people in this room are very familiar with what Pamrevlumab is. It's a monoclonal antibody. It inhibits the activity of CTGF, which we hope that it prevents fibrosis as well as inflammation. Our LYNX-1 study, this is the 093 study. This is the study in non-ambulatory boys, ages of 12 and older. This was a double-blind, placebo-controlled study, and it was Pamrevlumab, 35 milligrams per kilogram IV every 2 weeks. 99 patients were enrolled in this study. Our primary endpoint was change in pool at week 52. And then our secondary endpoints were pulmonary, cardiac, as well as change in hand grip. Okay. Unfortunately, earlier this month, we announced that our endpoint for PUL2 was not met. We are as disappointed as the community, patients, and we are fully committed at FibroGen to make sure that we share this data, full data, at an upcoming Congress, as well as we'll publish this results as well. Our LYNX-2 study, this is still ongoing, and in this particular study, this is ambulatory boys with between 6 and 12 years of age. Again, this is another randomized placebo-controlled trial, same milligrams of dose, 35 milligram per kilogram every 2 weeks. And this is looking at boys between 6 and 12, 73 patients enrolled. We hope to have this results, top-line results, in Q3 of this year. And what we're looking at in this particular trial is change in North Star at week 52. Secondary endpoints is change in forced air, climb velocity, 10-meter run, walk test, as well as time to stand, and finally, time of loss to ambulation. And that's it. Thank you.
Thank you so much. And now, Antisense Therapeutics.
Matt.
Sure.
And thank you to all of you who are here today. I see some familiar faces. I was on the board of directors at Sarepta for 8 years and attended these events, and so I'm thrilled to see many of you at this meeting. What I really want to do is maybe a wee bit different than the other presentations. I want to introduce you to our company because we're located in Melbourne, Australia. We've not been in the US, so you may not have heard about us. We've been around for a while, and we've actually conducted a study in Duchenne in Australia in nine participants. The data was very positive. I'll show you that now. A little more on the company. We are moving into a phase 2B study. In fact, we just enrolled our first patient in Turkey on June 6th. We're very happy about that. We've had to change a little bit. Initially, this bottom bullet where it says it had registration potential, it had registration potential in Europe. And so the initial design was to enroll 125 participants. Having said that, and as the result of some resource issues, we've had to redesign it and back up. So now the current study, same protocol, same outcomes that we're looking for, PUL2, cardiac, pulmonary, and quality of life. So same protocol, but in 42 participants. So you might ask, why aren't we, by the way, why aren't we in the US? So US FDA requires greater than 9-month tox data. We didn't have that. We're working on that now, and we'll have that tox data readout by the end of 2024. When that happens, we will, of course, come into the U.S. By the way, I've promised Pat that I'm going to be with you next year in Orlando for this meeting next year. One last thing on that. My colleagues from Melbourne would be thrilled to be here, but it was a little easier for me coming from Florida than it was for them coming from Melbourne. So what does our drug do? It turns down circulating CD49 cells and TGF-beta cells. So what happens as the result of that is it turns down, as Pat characterizes it, the downstream consequences of the disease, the inflammation and fibrosis. So the phase 2 data that I mentioned earlier in Melbourne, everything worked. We were thrilled by it. Pull 2 was stabilized or improved. There was, positive outcomes compared to the historic controls. The activity of CD49 cells showed up, so that it gave us a lot of confidence in the mechanism of action. And then this bottom bullet, the MRI data showed that it stabilized the percent of fat in muscle cells and preserved function. So again, all very positive. This is the data that has allowed us to move into this now phase 2B study with a protocol that has registration potential. So hopefully we'll have this readout in 2024 as well, have the 10-month tox data in 2024, which again will allow us to come into the United States. Again, very happy to see many familiar faces, and looking forward again, Pat, to being with you next year in Orlando.
All right, Larry. Larry Markham is going to talk to us about Ifetroban.
This remote looks pretty straightforward, a big green arrow. So I'm Larry Markham, I'm a cardiologist, and I'm at Riley Children's and part of IU Health and IU School of Medicine. And I'm talking about the FIGHT DMD trial, which is an FDA-sponsored study aimed to impact cardiomyopathy associated with DMD. I have no direct financial disclosures other than being the PI for the clinical trial. Ifetroban prevents fibrosis and inflammation by blocking thromboxane receptor signaling, and you can see the cascade of events after a DMD gene mutation and abnormal dystrophin, resulting in inflammation and fibrosis of the heart muscle, leading to dysfunction. In preclinical animal model of DMD mouse, survival, which is an important outcome, and cardiac output were improved. Safety is well established with nearly 1,400 clinical trial participants, not in Duchenne, but in other diseases or other conditions, focused on fibrosis in those disease entities. The tablet is a dime-sized, once-daily medication taken in a fasted state. And so the study was to evaluate efficacy, safety, and PK of Ifetroban in Duchenne, and 2/3 of the subjects were randomized to active drug. And so this is the outline of the trial design. Participants were divided into 2 cohorts. One would be an early stage, and when I say early stage, that's early stage for cardiomyopathy, where the ejection fraction was greater than 45%, and then late stage, from a cardiomyopathy perspective, those with an ejection fraction of 35 to 45%. Individuals were randomized, as I said, into 2/3 of treatment, and those were further divided into a low dose and high dose, and then followed for 12 months' time. You can see the study timeline, which shows October 2020 was the first patient enrolled, and then we have had safety updates at PPMD meeting, and we have started the interim efficacy analysis for the first 25 participants who completed 6 months of the trial. And the hope is that by the end of this year, we will complete the late stage enrollment. The enrollment for early stage participants has been complete, and those should finish the entire 12-month period later this year, if early next year. And this is to remind me that there have been some protocol amendments for the late stage participants, and so I'll go through those at the end. And so if you look at the baseline characteristics of the first 25 patients who had completed 6 months of treatment, you can see it's a nice mix of ambulatory versus non-ambulatory, ventilatory support versus not, and then, as expected, the division of early stage versus late stage. And then, given the trial design, it's always important to show that the different therapies and how they were allocated between the groups, and those were all relatively equal. When we look at the safety, these again were from the 25 participants at 6 months, and you see that there were no significant differences between high or low dose or even the placebo, meaning that patients with Duchenne have a lot of symptoms, and so we tracked all of those in all of the groups. The most frequent events were nausea, vomiting, an occasional viral infection, and then some headaches. At efficacy, again, there were no differences in quality of life scores among the participants or pulmonary function tests, and there was a lot of variability in the arm and leg QMT, maybe a trend in the low-dose group, but that data, given the large variability and small sample size, needs further analysis. And as stated, we're about halfway through all of the cardiac imaging biomarkers, so that data should be forthcoming relatively soon. And so in summary, both low-dose and high-dose Ifetroban are well tolerated in the first 25 DMD patients to complete 6 months of treatment. The interim analysis supports no significant changes in quality of life survey and pulmonary function test. Maybe a positive trend with low dose in the leg QMT. As I stated, the cardiac MRI analysis is in progress. The early stage cohort has met the enrollment goal and it is closed at present. And later this year, we'll have the 12-month completion for that cohort. And then the changes for the late stage involve really two protocol changes based on feedback from patients. And so those two changes are that the protocol, as you saw, had 3 MRIs, one at baseline, one at 6 months, and one at 12 months. We have made the 6-month MRI optional, however, suggested an echocardiogram for safety to make sure there was not a significant change in function. And then, as I stated, the initial protocol was for the ejection fraction to be between 35 and 45%, and that was our definition of late-stage cardiomyopathy. And so we broadened that to include if your ejection fraction was ever that low, even if your treatment has been modified, you have improvement over time, you would still qualify as being eligible for the late-stage cohort. So those candidates, again, whose ejection fraction has ever been that low, can still qualify for the study, and so reach out if you have interest. Again, I want to acknowledge and thank the participants and families who have been involved in the study, as well as all of the groups who are conducting the trial and the funding and support that we have received. So thank you very much.
All right, thank you all so very much. Questions? All right, Mindy.
Hi, I'm Mindy Cameron. What do you foresee with Ifetroban being the regulatory path forward if we were to continue to see, good results? So, how are we tying cardiac outcomes to function? I know this has been challenging. And then the second question is, for participants in Ifetroban, do you see the expanded access program continuing indefinitely, or what's that look like?
At least for now, we have amendments to the protocol which just extend that. So it's as of now, it's going to be up to 2 years, if not longer. And so that's ongoing conversations. I am not sure I can answer the regulatory and the Cumberland's plans as far as that goes. I will say our question from the previous session focused on carriers and is there interest or plans, and we have had conversations, or I have had conversations with Cumberland that if there is a signal at all in some of our MRI data, then I think we can really have tangible conversations about what is the timeline for considering a study with carriers. But Inez and Ingrid are here from Cumberland, so they would probably be able to answer the regulatory questions. Yeah.
Other questions? This is a quiet group. Other questions? All right, so Joanne, I'd love for you to talk about post-gene therapy studies. I'd love for you to tell us if this drug, how you give it, because I don't know that you mentioned that it's oral, so now I just said how you give it.
Mm-hmm.
But if you talk about combination therapies, how do you see this happening? When do you see it happening?
Mm-hmm.
And what is Edgewise thinking about this?
Mm-hmm. Yeah, thank you. I think that everyone in the field is really encouraged by seeing a lot of different therapies, multiple therapies coming along. And it's going to be a big question of how to fit these all together. I mean, we're already there in terms of already looking at drugs on top of steroids, of course. So we've already got two things. What we see in our trials is to include dystrophin-targeted therapies, so the multiple exon skipping therapies. Boys who have been on exon skipping therapies are already, if they are stable for a year, they can be enrolled in the Duchenne study. So we see it on top of that. The next logical thing is gene therapy. And so we are planning to add an additional cohort in our current phase 2 study that will enroll boys who have been on gene therapy. What we are doing in our current therapy trial, as I told you, is to look to make sure that we have the right dose for phase 3. So that's what we are doing first. We anticipate having data from the first 3 cohorts by the end of the year, and hopefully that will give us the information to choose a dose and to move on to patients with gene therapy as well as a phase 3 study. So I think that is, that is one approach for looking at that, because it's clear that there is going to be a need for multiple therapies. If we're talking about moving Duchenne into Becker, well, we're also developing in Becker because that's not great either. We need to treat that as well. So that's why we're looking at the whole spectrum of disease and how EDG 5506 can preserve, protect, and improve muscle function along that spectrum. What did I miss? You asked about 5
No, it's
questions.
good. Just one further question is that if you're thinking about post-gene therapy, I have 2 questions. First of all, and please interrupt me if you have. So if you're thinking about a post-gene therapy study, are you thinking about how many years you have to be out? Can I have gene therapy on Tuesday and 3 months later start on your drug?
Great question. And so one of the harder things in looking at investigational therapies is you want to understand what your investigational therapy is doing by itself. So if, for example, with exon skipping therapy, that's an effect that builds up over time. So that's why we ask for kids to be on a stable dose of exon skipping therapy for a year before they're enrolled in the study. With gene therapy, there's more to learn, isn't there? We don't know what dystrophin does over a period of time. And I think that as we see these data come out, we'll understand that. But I think it needs to be a period of time after gene therapy. So it's not Thursday when you had it on Tuesday. It's to get out past that time where the side effects of gene therapy are seen, and once the kids are relatively stable, so that we can see an effect of another drug on top of it and understand that better. Does that help?
Yes, thank you. Another question. Terry?
Thanks, Pat. This is for Joanne, but really anyone can comment. And I've asked this question the last couple of years and still haven't really received a solid answer because I think it's still being explored and investigated and murky. When would be considered that a Duchenne boy, young adult, after being on therapy perhaps for several years and holding stable, and some say should be considered Becker's, and then enrolls in trials, maybe for Becker patients, but the pathology though is really Duchenne. Mm-hmm. So is there any clear-cut answers on that yet? I was told yesterday in a session that, yeah, it's still murky, that's a question, but it is in discussion. Can anybody discuss that, or are we making progress?
Yeah. Maybe I can take a crack at it, and I'm happy to give it to anybody else. It is a continuum. It is a spectrum. There are differences in Becker, probably because if it has a later onset and puberty has already happened with testosterone effects, but it's clearly a continuum. The FDA guidance that PPMD is putting together tries to address that question, and I heard from the experts that was a very difficult question to answer. There used to be a definition that if you walked past 12, you were Becker. But then with current therapies, with steroids being instituted at a younger age, that's not true. And so now is it 16? But that suggests that somebody who loses ambulation at 15 and 9 months is different than at the day after their 16th birthday. So there is a continuum, and I think that it is helpful to look at these kind of therapies, like we're looking at something that's addressing contraction-induced injury that is true for Becker, it's true for Duchenne, to be looking across that spectrum. And that's what we're trying to do in clinical trials, to address a dystrophinopathy, not just Becker or Duchenne.
Yep,
so I completely didn't answer your question, give
no,
you an answer
did.
of it.
And it is sort of what was told to me yesterday. And I know Pat's working on it with her teams, because we are going to be seeing more boys transitioning, if they have success on the ASOs or gene therapy. Mm-hmm. But I was just wondering where we are with that.
Yeah.
So I can answer where we are with that. We're surveying the certified centers to ask them how they diagnose Duchenne or Becker, because some people diagnose it exactly as you said, walking into a certain age splits the difference. And some people talk about the in-frame, out-of-frame rule. Remember, in-frame, where there's the ability to express dystrophin, and out-of-frame, where there's no ability. And I was in a meeting once when somebody was talking about the in-frame, out-of-frame rule, except when it doesn't work, right, except 3 to 7 and this and that and the other thing, and those are making mistakes. And I was like, So I think we need better definitions. We did an ICD-10 code some years back to differentiate Duchenne and Becker from other things, and I think there's going to be more nomenclature around that. But we worry when people say we're going to change Duchenne to Becker, because sometimes if you change the diagnosis, then services are lost, and access to a medicine that's for Duchenne might be lost. So we have a lot of work to do around that area, but I think it's murky. Larry.
I was just going to say that every cardiologist who is here and engaged with PPMD, when we converse or when we talk, we have sort of began thinking of this as just dystrophinopathy, and we try to define the risk, meaning that I think we feel comfortable with a Duchenne timeline. We're reasonably comfortable that most Becker timelines from a cardiac standpoint are slightly delayed. But having said that, it's sort of an all-encompassing package from me for a cardiac risk, because if I had a Becker patient who was weaker than I thought they should be or could be, I'm going to say, well, your cardiac risk is higher than this 28-year-old who's still running a marathon and has a diagnosis of Becker. And so we're looking at the heart muscle and saying, that tells me how much to worry and how much the risk is, almost independent of the name or the label that's put on it. And I think now that's the clinicians. I think from a regulatory standpoint and a research standpoint, that has not transcended as of yet, but I think when we have therapeutics designed at ejection fraction, to me it becomes a moot point in kind of how you got there. That's where the term dystrophin can just come into play, and you can enroll all subjects and just focus on the cardiac outcomes.
Yeah, that's really an important point. Thank you. And I think going forward, talking about clinical trials, and Pat's point was important as well. You change over to Becker's, you lose services. But another segment of this is when eligible for clinical trials. Are we sticking with the original pathology of Duchenne, or because they did well on a drug, they're really maybe Becker's at age 20? Yeah. That's something that needs attention as well. It's like, where do you draw the line?
A lot of attention, yeah.
Yeah, we have to get there, and maybe there's some day that trials will be based on characteristics, not on this name or another name, but based on a certain number of characteristics. And I think the consortia are trying, the CTAP consortia and the D-RISC consortia, both of those consortia are looking at data to see if we can get there. So the inclusion criteria are, maybe certain functional measures, PUL versus North Star versus other measures, and those people are included in this study, so I would hope so.
Thank you.
Thank you. So Larry, while you have the microphone, here's what I want to know.
Would you like some water?
I think I'm okay.
Okay, all right.
What I want to know is, people are on cardiac medicines. People are on ACE inhibitors or ARBs, and they're on eplerenone, and if their heart rate gets too high, sometimes they're on a beta blocker. How does this drug fit into that system?
Yeah, so that's one thing that all of us who were involved in the creation of this trial were pretty adamant about, that this is an ancillary or on top of medication. And so any standard of care medication that has been proposed in Duchenne is allowed in this trial. So if you're on 4 drugs from a heart standpoint, and they're all FDA-approved therapies for known cardiac failure or dysfunction, come on, it's still good. Now, I think some of the other things that may still be investigational or non-FDA approved, or you've just been in another therapeutic trial, we exclude those patients, but it's for any known and approved cardiac therapy. So this is on top of, so that we have real-world data of how to use this once the study's over.
Great, thank you. So Linda, you are thinking about including, or really going to include, post-gene therapy folks. And so two-part question. If you're on an Antisense and want to go into Capricor, how long is steady state? How long do you have to be on the Antisense before you start Capricor? And when you're thinking of a post-gene therapy study, is it gene therapy today, Capricor tomorrow? Tell me what you're looking for as well.
Yeah, so thank you for giving me this opportunity. Actually, I was corrected by my clinical team. We are currently taking post-gene therapy patients into HOPE-3 as long as they are 1 year post-gene therapy, and that's because obviously we have to sort of let everything even out. In terms of Antisense, the same basic parameters, anything that's already approved, as long as they're on a stable dose and they haven't had any thing beyond a weight-based adjustment, we're taking all of those standard of care patients into HOPE-3. In terms of anything that's investigational, there has to be a washout period. It depends on what the drug is. That's sort of reviewed by the clinical team and evaluated based on the washout period and that kind of thing. So we've been getting a lot of requests about that, especially patients that would be similar to our patient population that were recently in FibroGen. So we have to wait for the washout, and then of course we'll welcome you into HOPE-3 and anything beyond.
Okay. One last question about that. So in the last session where we did the same companies, what we also talked about is someone got up and said, my son is in Capricor and he gets this bump and then he starts to slide a little bit before his next bump. So he can feel the difference in his body, and then obviously once he gets the next, he feels better. So you said you all are thinking about amending your protocol to perhaps make this a more frequent dosing schedule than quarterly. Can I ask, when you thinking about altering that protocol to amend it? I know you won't amend it within a phase 3, but when do you think you'll amend it?
So, I think everybody is aware of the fact that the greatest bar we have to overcome is to get our therapies or medications approved by whatever regulatory body. For us, it's the Food and Drug Administration. We need to get across that line. So HOPE-3 is a really powerful study because it capitalizes on everything that we learned in HOPE-2. The open label extension data is a great guidepost of the potential for CAP-1002 in treating Duchenne. We saw a 2.2 decline in PUL scores over a 2-year period where standard average decline is 4 points. That is very good. The every quarter dosing was based on practical reasons, based on preclinical as well as the HOPE-1 study published in Neurology. Once we get approved, I think it's obvious that we want to indication expand to the younger kids as soon as we possibly can, and then explore more frequent dosing. And I think that would probably be something that would happen post-approval, and we're laser focusing on how to get that done as quickly as possible.
Good, thank you. And I can't, Dr. Patel, would you talk to us about, you mentioned you're going to get the phase 2, the secondary, the exploratory measures and the secondary outcomes, and you're going to present that data at future conferences. You're actively now reviewing that non-ambulatory data, and can you give us a timeline about when you could say something about that?
Sure, absolutely. In terms of the 093 Lelantos 1 non-ambulatory study, obviously, our PUL-2 data did not hit its endpoint. However, we are in a process right now evaluating the cardiac, pulmonary, as well as grip strength, and we are committed to publishing this data and hopefully being able to present it at upcoming Congress. In terms of publication, that takes a little bit, but we are planning on publishing the entire data.
And do you have a timeline that you'll be talking about the data or not?
It would be great to be able to get into the next medical congress, right? The next one's up is WMS, so that was our initial intent, was to get up into
World
be able to
Muscle
have
Society.
at least an abstract oral presentation at WMS.
Okay, and your ambulatory study reads out?
Yes, it reads out in Q3 of this year, so top line data should be out within the next quarter.
Good, thank you. All right, and Gil, one other question. You said once you get the tox data for the FDA, you're going to be, will you open studies in the U.S.? Is that the plan?
That is indeed the plan. We want to be here. We're in Europe because of regulatory reasons. We're not in the U.S. because of regulatory reasons. So as soon as we cross that threshold we will be here. By the way, I did want to point out that the study that we're currently conducting is in the non-ambulatory population.
And do you think that's where you'll go as well for a
Yes.
study in the U.S. in a non-ambulatory population?
Yes.
Will you include antisense oligonucleotides, exon skipping? I know you're antisense oligonucleotide, but just to kind of draw the line, if a person is on one of the approved products that are approved in the United States, will this be an additive drug in your view?
It absolutely will. That's how we view it. It will be accretive to those other drugs, those other therapies.
Okay, thank you so much.
Thank
Joanne,
you.
one last, sorry, did
Thank
I cut
you.
you off? Okay. Joanne, one last question for you. In that Becker study, how young Becker and how old Becker? What's the age range?
So for Becker, we have been enrolling age 12 to 50. For Duchenne, 4 to 9. But that's really on functional criteria we are. And the idea being that when we're looking at dystrophinopathy, we are spanning the age range. And we're also spanning without steroids in the case of the Becker folks and with steroids. So we're trying to understand the entire spectrum of disease. So.
Okay, and so then you're recruiting the Becker patients. That's a wide age range, considerable functional variability, I'm guessing. And what's the primary here? What are you looking?
Yep.
Because we are obviously well aware of primary outcomes and how they can say yes or no to progress. So tell us what your primary is in the Becker.
So for Becker, we're enrolling a group of patients that are ambulatory and that have North Stars between 5 and 32. And that's because the decline in that group of patients is relatively homogeneous. So that we can power a study appropriately. So that's the group we're enrolling. That's not to say that we wouldn't anticipate seeing benefit in a broader range of populations. And that's also why we are looking at biomarkers in that population to support that we would see safety and an effect in that broader population.
Okay, thank you so much. Other questions? All right. You do. It's
for the virtual people.
Keeping the blood rolling in a cold room.
My question is for the Edgewise. Well, I guess for all of you, 2 questions. One is that the stable dose of steroids, does that include weight management adjustments? And
Yes.
So you can make those and that still is considered? Okay.
It's mg per kg.
Perfect. And I forgot my other question, so I'll come back to you and find you.
And I'll just ask a really quick question to Edgewise as well. For the phase 3 trial that you said you were going to do, because phase 2 is being set up for a phase 3, what would the difference be? Would there be a different dose or anything?
So we'll choose a dose for the phase 3 study, and then that study would be a functional endpoint over whether it's 12 months. And because the FDA, for full approval, is looking for a functional endpoint, they're not quite there on biomarkers at this point. So,
I remember. Any wiggle room with the 95 fifth percentile BMI for the Edgewise? I've heard.
we took that out with an amendment.
So there's no BMI requirement.
So I had a comment about endpoint. So, because the study is supported or funded through the FDA, and John Soslow, who I can see his slides right there, so that's the next session after lunch, has had conversations with the FDA. And one of the things, and then in follow-up, so if you want a cardiac approval for a drug, the outcomes usually are mortality and heart failure hospitalizations. And ejection fraction is not exactly an outcome measure per se, according to the FDA. And so it can be. But we just had a paper that came out in Circulation Heart Failure last month, which looks at trying to quantify mortality risk from a cardiac standpoint in Duchenne. And our hope is that that paper then becomes the ground floor of these are measures that can be associated or predict mortality in Duchenne cardiomyopathy. And so that data can be used by anyone who's using cardiac outcomes short of hospitalization and heart failure admissions. So we're working on it.
Yeah, thank you for that, because it's really hard to get things understood by the renal cardiac folks over there at Center for Drugs. So I think that's really a wonderful paper to at least give them food for thought to be thinking about that. Any last questions? Tally, did you have a question, or was that? Okay. All right, so it's 1 o'clock. Box lunches are outside. Thank you all.
Thank
we'll
you,
hold
Pat.
you to those trials. Thank you so much.